Welcome back. In the last section of the course, we discussed the general organization of animal bodies. We talked about principles of regulation, how homeostasis is maintained by negative feedback loops. And now we're going to go on to think about the major themes in this course. One of them is the transmission of information across large multicellular bodies. And the second is the ability to respond to the environment. So remember, these are two of the things on that list of things that all living organisms do. And those are the two that we're going to be focusing in on for the rest of the semester with regards to animals. So we've already looked at many of the ways that plants can respond to their environment, to different stimuli. We thought about how they can respond to touch or pressure, how they can respond to gravity, how they can respond to light. And so now we're going to think about animals in the same sense, and obviously there are many different kinds of stimuli in the environment that animals are capable of responding to. We know about all of the different senses we have, and so now we're going to start to think about how animals go about responding to things in their environment. And we've already looked at some of the different ways in general that information can be transmitted through a multicellular body. We talked about paracrine secretion, which are chemical signals that are going to be sent to very nearby tissues from neighboring cells that are going to cause some kind of response in a target cell. Remember, this is a very specific short distance communication. We talked, especially with regard to plants, about hormonal regulation, hormonal transmission of information system-wide through these chemical messengers. In the case of animals, these are going to be carried by the blood throughout the system, and this is going to be more long-distance transmission of information, but fairly non-specific. It's going to be blasted out to the entire organism, and the tissues that respond are going to be the tissues or the cells that have the particular receptors for that hormone. But it's going to be a rather broad, coordinated kind of signal. When we look at the nervous system, remember that we're talking about long-distance transmission of information, but targeted to very specific cells. So this projection from the neuron is going to transmit that signal just to the cells that it makes connections with, and so this is going to be a long distance but highly specific form of communication throughout a large multicellular body. So let's think in more detail about this nervous communication. So neurons are nerve cells that transfer information within the body. Don't get confused with the term nerve. A nerve is actually a bundle of neurons that travel together to similar destinations, but the neuron is the actual cell, the nerve cell. Neurons use two types of signals to communicate. So they're going to use electrical signals to transmit information over long distances. Once that electrical signal reaches its target, it's going to use chemical signals to transmit that information over the short distance between the end of the neuron and whatever its target is. So two modes of communication are going to be used by neurons. Interpreting signals in the nervous system involves sorting a very complex set of pathways and connections. So any individual neuron can be getting information from many different directions, and the pathway it's going to take is going to determine the kind of response that it gets. So really understanding everything that's going on in the nervous system is a very, very complicated problem. Processing of information takes place in either simple clusters of neurons called ganglia, or a more complex organization of neurons called a brain. So if we think about the different kinds of nervous systems that we find in animals, we can have anything from this kind of diffuse system of nerve cells called a nerve net, and this is what we see in simpler animals such as cnidarians like this hydra or a sea anemone or a jellyfish. We're just going to see this kind of spread out network of nerve cells. and we call this a nerve net. In animals that need a little bit more coordination of their activities, we start to see bundles of these nerve cells coming together to form ganglia that are going to be integration centers for information processing to coordinate the activities. So here we see a couple of clusters of neurons in this flatworm forming ganglia. In much more complex organisms that really require a lot of complex processing of these neural pathways, we see a large cluster of neurons that are going to be involved in information processing, and we call this kind of arrangement a brain. And so we see these in arthropods, we see these in all vertebrates, in any organism that's going to have any kind of complex movement or behavior. So neurons are very highly adapted for this function of information transfer. So let's take a look at how a neuron is put together. So the neuron is a cell that exemplifies the close fit between form and function. So we have a couple of different regions of a neuron. Most of the neuron's organelles are located in the cell body, so it's going to have the function of doing all the business of keeping the cell alive. Most neurons have dendrites, which are highly branched extensions that are going to have the function of receiving signals from other neurons. So these are going to be kind of the input centers, and having this highly branched structure is going to allow the neuron to receive input from a lot of other neurons. And then there's this transmitting region called the axon. So the axon is typically a much longer extension that's going to transmit the signal to other cells at synapses through an enlarged end region called the axon terminal. And then the cone-shaped base of an axon where it connects to the cell body is called the axon hillock. The term synapse refers to a junction between the axon terminal, so the very end of the axon, and the other cell that it's communicating with. So the synapse is actually a space in between. The synaptic terminal of one axon is going to pass information across the synapse in the form of chemical messengers. So this is that short distance chemical transmission that we were talking about just a moment ago. And these chemical messengers are called neurotransmitters. So here we can see a diagram of what I just described. So we can see the cell body. This is where the nucleus and all the mitochondria and everything that are necessary for the cell to remain alive are located. We can see the dendrites that are these little projections that are going to branch out and make lots of connections with other neurons. The axon hillock is this sort of enlarged region here where the cell body meets the axon, and the axon is going to be this long projection that's going to make contact with another cell. So we call this neuron here, with reference to this connection, the presynaptic neuron, and then this one would be the postsynaptic neuron. So obviously this is also going to be a postsynaptic neuron for some other neuron that's making contact with these dendrites, but with respect to these two, presynaptic cell, postsynaptic cell. And as we see here, in this connection between the presynaptic cell and the postsynaptic cell, the mode of communication is going to be through these neurotransmitter chemical molecules that are going to be released into that space, the synapse, between the two. So the general flow of information through a neuron is going to be in through the dendrites, being integrated in the cell body, although axons can also connect directly with the cell body itself, not just the dendrites. So a lot of times there'll be synapses directly on the cell body, but in through the dendrites, through the cell body, and then out through the axon, communicating that message to a target. And that target can be either another neuron, so as in this case with the presynaptic and postsynaptic neuron, it can be just transmitting that information to the next neuron in the pathway. It can be a gland. That message can be telling a gland to release some kind of hormonal signal to coordinate a full body response to whatever the stimulus is, or the target can be a muscle, signaling that muscle to contract and create some kind of motor response. And as you can see here in this image, these neurons are going to form these dense networks with lots and lots of communications with lots of other cells. And so these neural pathways can be incredibly complex. In addition to these actual neurons in the nervous system, we see lots of these support cells known as glia or glial cells. So most neurons are nourished or insulated or physically supported by these glial cells. Now let's take a look at how the vertebrate nervous system is organized. In the central nervous system and the peripheral nervous system, those are the two major divisions of the vertebrate nervous system. The central nervous system, or CNS, consists of the brain and spinal cord. The peripheral nervous system, or PNS, can be divided into two subsections. One is the sensory part of the peripheral nervous system. We call this the afferent portion, afferent with an A. Again, this is very unfortunate terminology because there's only just a very small difference between these terms. The other component of the peripheral nervous system is the motor or efferent part of the peripheral nervous system. So the sensory nervous system can again be subdivided into two categories. One is special sensory, and so these are the senses generally associated with the head invertebrates: smell, vision, hearing, and taste. And then general sensory, associated with the rest of the body, touch, and proprioception, which is a sensation that allows your body to detect when muscles are being stretched or other tissues are being stretched. The motor or efferent side of the peripheral nervous system can also be divided into two subsections. The somatic nervous system is going to communicate with voluntary skeletal muscles, so the striated muscle that we talked about when we were talking about the different kinds of muscle tissue. And so this is going to be the part of the motor nervous system that you have conscious control over. The other side of it is the autonomic nervous system. The autonomic nervous system can also be broken down into two categories. One is the sympathetic nervous system, and this is responsible for the fight or flight responses. And then the other part of the autonomic nervous system is the parasympathetic nervous system, and this is the part that is involved in maintaining homeostasis, the rest and digest. So these are kind of opposing components of the autonomic nervous system. So now let's take a look at the way information flows and is processed in the nervous system. So nervous systems process information in three stages. Sensory input, so information is going to come in from the outside world. Integration, so it's going to be processed in some way to determine what should be done with that information. So this is going to be the job of the central nervous system, the CNS. And then that's going to result in some sort of hopefully appropriate motor output in response to that sensory input. So three stages: input, integration, and then output. So sensory neurons are going to transmit information about external stimuli such as light, touch, smell, any of the senses that we use. This is an example of a sensory neuron. They're a little bit unusual in that the cell bodies are located somewhere along the axon rather than being in between the dendrites and the axon. Then the sensory neurons are going to make contact with one or more interneurons. These are going to be the processing part of this. These are going to be located in the central nervous system, and they're going to relay that information into the right centers of the brain or the spinal cord to determine what the appropriate response ought to be, which pathway that information ought to take. And so this looks more like what we already diagrammed a typical neuron to look like. We've got lots of dendrites that are projecting off of the cell body here in black. And in the case of interneurons, the axons are going to have lots and lots of projections to make contact with lots and lots of other neurons. And then finally, there are going to be motor neurons that are going to transmit those signals to muscle cells. Actually, could also transmit them to glands, but we call them motor neurons because most of the time they go to muscle cells, causing them to contract to create that motor output, that response. And so for motor neurons, we see lots and lots of dendrites taking in lots of information from lots of interneurons, a long axon that's going to be targeting a very specific set of muscles that it's going to activate. So this pathway of sensory processing can best be exemplified by something called a reflex arc. So what we see in a reflex arc is that some sort of stimulus is detected, in this case touching a hot candle. That information is going to be taken by sensory neurons toward the central nervous system. So this is part of the peripheral nervous system, outside of the brain and spinal cord. You can pick up that sensory neuron coming here toward the central nervous system. It's going to have its cell body, actually before it gets to the central nervous system, along that long axon, and it's going to communicate with an interneuron. So in the case of a simple reflex arc, this is going to be something where a very quick response is needed, and actually all of this processing in this case can happen right here in the central nervous system. When there's more complicated processing required, this is going to move through the spinal cord. So this is just a section through the spinal cord up toward the brain. The brain will process it and send a message back down through the motor neurons. But in this case, this is a simple reflex arc. There's going to be an interneuron that's going to make contact with a motor neuron here in blue that's going to come back out of the central nervous system, out of the spinal cord, make contact with a muscle. It's going to tell that muscle contract to quickly move that hand away from the source of that heat. And this is the kind of pain reflex that we see, completely involuntary because it's happening at the level of the spinal cord, but a very simple reflex arc that goes from input to processing in the central nervous system, in this case the spinal cord, to motor output. So how do these neurons work? Now that we have a better idea of how they're put together, let's look at their function. So this is going to come back to this idea of ions and ion pumps. So ions, as we know, carry an electrical charge. Remember, the definition of an ion is an atom or molecule that has lost or gained one or more electrons and has an electrical charge. So the cytoplasm and extracellular fluids adjacent to the plasma membrane contain electrically unequal distributions of ions. And this difference in charge creates what's known as an electrical potential across the plasma membrane, known as the membrane potential. So the membrane potential just refers to that difference in charge from one side of the membrane to the other. Membrane potentials refer only to a separation of charge immediately adjacent to the plasma membrane. So this potential is going to disappear as you go farther away from the membrane, either inside or outside the cell. So there's no charge separation slightly farther from the membrane. The whole solution taken together is going to be electrically neutral. There's going to be overall the same number of positive and negative charges, but immediately adjacent to that membrane, there's going to be a difference in charge. So just some terminology that we use when we're talking about this potential. The volt is a standard measure of electrical potential, and membrane potentials are measured in millivolts. They're very small charges, which is 1/1000th of a volt. By convention, membrane potentials are expressed in terms of inside relative to outside. So what that means is the outside value is defined as 0 millivolts, and however the inside is different from that is going to be positive or negative, depending on whether it's more positive inside or more negative inside. But the outside is always going to be defined as 0 millivolts. Generally, more negative ions are on the inside surface of the plasma membrane than on the outside. So, membrane potentials are usually negative. When a membrane potential exists, the ions on both sides of the membrane have potential energy. So remember, there's both a concentration gradient of these ions and also a voltage gradient that's going to, if they are able to cross the membrane, is going to cause them to move. So this is potential energy. They're held apart. They're prevented from moving by the plasma membrane, but if the membrane wasn't there, they would move. So if the membrane were removed, ions would spontaneously move from the area of like charge to the area of unlike charge. And this movement of ions, this movement of charge, is called an electric current. So ions move across membranes in response to concentration gradients as well as these voltage or charge gradients. And so the combination, remember we've talked about this before, of an electric gradient or a voltage gradient and a concentration gradient is called an electrochemical gradient. So how do we get this membrane potential, this kind of default resting potential? So when a neuron is not communicating with other cells, the difference in charge across its membrane is called the resting potential. So this potential exists because the interior of the membrane has relatively low concentrations of sodium and chloride ions and relatively high concentrations of potassium and some organic anions. And in the extracellular fluid, sodium ions and chloride ions predominate. So you can see that here in this image. We can actually measure this difference in potential using very small microelectrodes that are placed inside the cell to measure that difference in charge between the inside and the outside. So if each type of ion diffused according to its concentration gradient, organic anions and potassium would leave the cell along their concentration gradient, and sodium and chloride would enter. Let's take a look at how this resting potential is maintained. So ions such as these can cross plasma membranes efficiently in only two ways. So the first way is by primary or secondary or co-transporter active transport against a gradient. And so this comes back to our old friend, the sodium-potassium pump, that's going to be the active transporter of sodium and potassium across the membrane that maintains that resting potential. So the other way that ions can move across the membrane is by diffusion along their electrochemical gradient through an ion channel. So a protein that forms a pore in the membrane through which specific ions can diffuse. So these can either be passive channels that are open all the time, or they can be gated in some way that lets the ions pass through only under certain circumstances. We'll talk about that more as we go. So let's think again about this sodium-potassium pump and review how it works. So remember that sodium-potassium ATPase, that's the name of the protein that we call the sodium-potassium pump. So this protein is going to actively pump sodium ions out of the cell and potassium ions into the cell by using the energy gained from receiving a phosphate group from ATP. So let's just go through this whole process one more time. I'll also repost the little video that we watched in class when we talked about the sodium-potassium pump earlier in the semester so that you can see this in motion. But remember, in the resting configuration, the protein has a high affinity for sodium ions. When 3 sodium ions attach to it, then it develops an affinity for ATP. ATP is going to be phosphorylated and a phosphate group attached to this sodium-potassium ATPase. The bonding of the phosphate group is going to change the configuration of this protein, cause it to have less affinity for sodium ions and greater affinity for potassium ions outside the cell. So the sodium ions are going to be released to the outside of the cell, and now we've got a high affinity for potassium. Our phosphate is still attached, so it remains in this configuration. Once 2 potassium ions bind to this protein, that's going to trigger the release of that phosphate group. The release of the phosphate group is going to cause the change in shape back to its original configuration. It's going to decrease the affinity for potassium ions. It's going to change the shape of those binding sites, causing those potassium ions to be released to the interior of the cell. And now there's a strong affinity for sodium ions, and we're going to go back and repeat this cycle. So it uses 1 ATP for every turn of the cycle. For every turn of the cycle, we bring 3 sodium ions out of the cell and 2 potassium ions into the cell. And remember that this both creates a concentration gradient of sodium and potassium. It also creates a voltage gradient because we're removing more positive charges from the cell than we're bringing back in. We're removing 3 positive charges to the outside of the cell for every 2 positive charges we bring back in each cycle. So the pump establishes an electrical gradient as well as a concentration gradient. So the outward movement of 3 positive charges and the inward movement of 2 positive charges each time makes the interior less positive, more negative, relative to the outside. So remember, this is always relative to the outside of the membrane. And I just wanted to remind you of this activity that we did in class where we worked on the sodium-potassium pump. And so remember that if we're talking about flow of potassium ions, so remember this K here in the middle reminds us that we're talking about the flow of potassium. This dashed line represents the cell membrane. So above we have the outside of the cell, below we have the inside of the cell, and we've got an active flow of potassium from the outside of the cell to the inside of the cell, and this is going to cause a high concentration of potassium inside, a low concentration of potassium outside. This combined with the relatively high flow of sodium out of the cell is going to result in a relatively negative charge inside the cell, a relatively positive charge outside the cell, which is going to create a voltage gradient. Remember also that we have some passive channels for potassium, and this voltage gradient that we see in blue is going to tend to make the potassium ions want to flow into the cell, but the concentration gradient that we see in red is going to tend to make them want to flow out of the cell. So there are these opposing forces working particularly on potassium that is going to cause them to move into or out of the cell. So what we have are these passive potassium channels, what we call leak channels, that are just always open in the cell. So at rest, the plasma membrane of a neuron is relatively impermeable to most cations, but they have lots and lots of these potassium ion leak channels that allow potassium to leak across the membrane passively. So the concentration gradient favors the net diffusion of potassium out of the cell. So as the potassium ions move out of the cell, the inside becomes even more negatively charged relative to the outside, and the buildup of this negative charge is going to begin to attract the potassium and counteract that concentration gradient, keeping the potassium ions inside the cell. And eventually, the membrane is going to reach a voltage where there is equilibrium between the concentration gradient that moves potassium out and the electrical gradient that moves potassium in. So remember, at equilibrium, an equal number of ions are moving out as move in. And this voltage that results at the equilibrium point is called the equilibrium potential for potassium ions. And at this point, there's no net movement of potassium ions across the cell membrane, but remember, they're still moving just at equal rates in and out. And we can measure what this equilibrium potential is using an artificial membrane with potassium channels in it, and this is described by the Nernst equation. Don't worry too much about being able to use this, but we can figure out what this equilibrium voltage will be using the relative concentrations on either side of the membrane, and we find that the equilibrium potential for potassium alone is negative 90 millivolts. So once this reaches equilibrium in this resting system, the difference in charge inside to outside is going to be negative 90 millivolts. Even though chloride ions and sodium ions cross the plasma membrane much less readily than potassium ions do, each type of ion has its own equilibrium potential depending on the concentration difference and how leaky the membrane is to that particular ion. So for example, we can compute a separate equilibrium potential for sodium, and that will be plus 62 millivolts. So remember that the concentration gradient and the voltage gradient are both going to be driving sodium into the cell, so there's going to be a net flow into the cell causing a positive equilibrium potential. And so the overall membrane potential of the neuron at rest is going to be the combined effect of all of the individual equilibrium potentials of all of the different ions that are present. And so when we take all of these ions taken together with their equilibrium potential, what we find is that the equilibrium resting potential of the neuron as a whole is approximately negative 70 millivolts. So inside the cell has more negative charge, as we saw, due to the sodium-potassium pump than the outside of the cell. And this resting potential is going to be approximately negative 70 millivolts for most neurons. So let's take a look at what happens when that resting potential is changed by different events that happen, other neurons communicating with that neuron. When there's a difference in voltage between the two sides of the membrane, we say that that membrane is polarized. So this negative resting potential indicates a polarized membrane, and any change to that state of polarization we refer to as either a hyperpolarization, it's become more polar, the difference between the two sides is greater, or a depolarization, reducing the difference in voltage between the two sides of the membrane. So for example, when gated potassium channels open, potassium ions diffuse out, making the inside of the cell more negative. So that's going to increase the difference between inside and outside. So we call this a hyperpolarization. It's an increase in the magnitude of the membrane potential, an increase in the magnitude of the difference in voltage between inside and outside the cell. Opening other types of ion channels can trigger a depolarization, a reduction in the magnitude of that voltage difference between inside and outside, and we call this a depolarization. For example, depolarization occurs if gated sodium channels open and sodium ions diffuse into the cell. And so when other neurons are communicating with this neuron that was at rest, we get what are called graded potentials. So graded potentials are changes in the polarization where the magnitude of the change varies with the strength of the stimulus. That's what we mean by graded. A stronger stimulus is going to create a stronger change in the membrane potential. So if a depolarization shifts the membrane potential sufficiently, it results in a massive change in membrane voltage called an action potential. So here we see the resting potential at about negative 70 millivolts. We can see a series of graded potentials that are going to depolarize that membrane towards some threshold voltage. Once that threshold is reached, there's going to be a huge change in the membrane potential, and this is an action potential. Action potentials have a constant magnitude, so for this particular neuron, every time it gets to this threshold, it's going to completely depolarize and then become positive up to somewhere around plus 40 millivolts or so, and it's going to be the same change in the polarization of this membrane every time there's an action potential. So a constant magnitude. They're all or one. Once this threshold is reached, it's going to do this or it's not going to do this. There's nothing in between. And it's going to transmit that signal over long distances. Once that action potential is reached, it's going to be transmitted all the way along the length of the axon. These action potentials arise because some of these ion channels are voltage gated. So once this threshold voltage is reached, it's going to trigger the opening of these ion channels. And we'll look in much more detail at how this works in the next web lecture, which is specifically going to deal with action potentials. So how do these changes in membrane potential come about? So they come about due to these chemically gated, or what we call ligand gated channels. So remember at this axon terminal, the synaptic terminal, there are these chemicals called neurotransmitters released into this synaptic space, and they're going to be received by receptors on the postsynaptic cell. These receptors are going to respond to the bonding of these neurotransmitters by opening these ligand-gated channels. So the ligands can be hormones or they can be neurotransmitters, and when they bind, so here is our ligand. Remember, a ligand just refers to any molecule that's going to bond with a receptor in this lock and key kind of fashion. And so this is the ligand for this particular receptor. This is the neurotransmitter acetylcholine. When this fits into its receptor, it's going to open up this channel and allow sodium to flow into this postsynaptic cell. So we're looking at the postsynaptic cell here. This is the neurotransmitter coming from the presynaptic cell through this synaptic cleft, the space in between, and when it binds, it's going to allow sodium to flow through. and that's going to cause the membrane in this region to depolarize. Positive ions are going to come in. The difference between inside and outside is going to decrease. So it's going to open ion channels and allow diffusion along this electrochemical gradient. So this can have a couple of different effects on the postsynaptic cells. So here we have neurotransmitters that are opening up sodium channels. And so those sodium ions are, of course, going to flow in to the cell along both their voltage and their concentration gradient, depolarizing the membrane in this region. And so from our resting potential of -70, we're going to see a little depolarization as a result of the activity of that neurotransmitter. And we call this an excitatory postsynaptic potential. So when it's depolarized, that means that it's going to make this postsynaptic neuron more likely to reach that threshold and fire an action potential. But this can also open other kinds of channels. So a different neurotransmitter might, for example, open up chloride channels. Chloride is going to flow along its concentration gradient into the cell. And so when these open, chloride flows in. And so now we're bringing more negative charge to the inside of the cell, and that's going to hyperpolarize this membrane. It's going to make the inside more negative. And this is going to make the postsynaptic neuron less likely to fire an action potential. So we call this an inhibitory postsynaptic potential. It's going to make it less likely to fire. So these different postsynaptic potentials are going to be coming from different neurons, and they're going to add together. So the cell body of a postsynaptic neuron is going to be the integrating center that's going to receive all of these signals and sum them together to make a decision about whether to fire an action potential or not. So the cell body may receive inputs from hundreds or thousands of different synaptic terminals coming from different pathways. So a single excitatory postsynaptic potential is usually too small to trigger an action potential in a postsynaptic neuron. It needs to get that message either at a very high rate or from a lot of different neurons that are communicating with that neuron. So individual postsynaptic potentials can combine together to produce a larger potential in a process called summation. So if two excitatory postsynaptic potentials are produced in rapid succession in time, so one right after another, an effect called temporal summation occurs. So summation in time is what temporal refers to. So if they come very close together, it's going to increase the magnitude of that change in membrane potential. In spatial summation, excitatory postsynaptic potentials produced nearly simultaneously close together on the neuron by different synapses are also going to add together. So if they come close together in time, and in different regions of the cell, they're also going to have this additive effect and increase the amount of depolarization or hyperpolarization that's produced. So the combination of excitatory postsynaptic potentials through spatial and temporal summation is what can trigger an action potential. It's going to take a bunch of these signals. So just to see this a little bit more visually, so here we have two separate excitatory postsynaptic potentials that are coming not very close together in time, and you have a little bit of a depolarization and then a repolarization, then the next signal comes, and then the same small depolarization, then repolarization. It's not going to reach that threshold for an action potential. So this is a case where there's no summation and you're not going to get there. If, on the other hand, signals are coming through one neuron very close to each other in time, one after the other, there's going to be one excitatory postsynaptic potential. It's not going to come all the way back down to the resting potential before the next one comes. So it's going to start from this point and go up. And that could push it up to this threshold for an action potential just because the second signal came along before the effect of this first signal had gone away. So this is that temporal summation. You get a larger effect because they're coming through in rapid succession. Here's an example of spatial summation. In this case, we've got excitatory postsynaptic potentials coming close to the same time in different regions of the neuron. And so the two of them come together and their combined depolarization could get it up to the threshold to produce an action potential. Excitatory and inhibitory postsynaptic potentials can also interact with each other in an additive way. so that the effect of an inhibitory postsynaptic potential can negate the action of an excitatory postsynaptic potential and make it less likely that the threshold potential is going to be reached to produce an action potential. So looking at this a slightly different way, so imagine we've got an excitatory postsynaptic potential here that causes a change, a depolarization of about 1 millivolt, and then the next one comes along, maybe more neurotransmitter is released or a different kind of neurotransmitter is released, so that this excitatory postsynaptic potential creates a change in membrane voltage, a depolarization of 2 millivolts. Okay, so these are coming separately, they're repolarizing before the next one comes along. Maybe the next one is an inhibitory postsynaptic potential that's going to hyperpolarize the membrane by 1 millivolt. So these would be the effects that each one of these would have separately. If they all came in together, they would sum together. So you would have the net effect of this 1 millivolt depolarization plus this 2 millivolt depolarization. So you'd have a total of a 3 millivolt depolarization, but you'd have to take away this 1 millivolt hyperpolarization. It's going to interact with that in an additive way. So the net depolarization, if all three of these came in together, would be a depolarization of 2, because basically this inhibitory change of 1 millivolt is going to cancel out this excitatory change of 1 millivolt. So again, the cell body is going to be where all of these different signals coming from all of these different inputs, these different neurons that are communicating with this neuron, and sum all of these inputs together. So as the inhibitory postsynaptic potentials and excitatory postsynaptic potentials are received throughout the dendrites and the cell body, the charges are going to spread to the axon hillock, and that's going to be where an action potential is going to be initiated if the cell as a whole reaches that threshold potential. So a good analogy for this might be skydiving. So if you think of a skydiver in the plane deciding whether it's going to go or not, it might have a lot of friends on the plane that are yelling, jump, jump, do it, do it, do it. So these would be the excitatory postsynaptic potentials. Might have another group of friends that are on the plane yelling, don't jump, don't jump, don't do it. So these would be the inhibitory postsynaptic potentials. But once our little squirrel skydiver here hears enough of these jump, jump signals, it's just going to either jump or not jump. If it jumps, it's going to jump all the way. So this all or nothing property of action potentials is very similar. Either the action potential is going to go or it's not. And it's going to be based on basically how loud these signals are and how much they cancel each other out. So if the net signal is go, fire, jump, do it, then it's going to fire, and it will do it in this all or nothing kind of way. So if the membrane at the hillock depolarizes past the threshold potential, an action potential begins, and it's going to be propagated down the axon to the next synapse. So it's going to transmit that message. So in this web lecture, we have gone over the structure of a neuron, how it is put together, how its form dictates this function of information transfer. We've talked about how resting potentials are generated and maintained by the sodium-potassium pump, creating this electrochemical gradient that's going to have an equilibrium, a net equilibrium potential of around -70 millivolts. So that's the resting potential. We've seen how this resting potential can be changed by inputs coming from other neurons in this graded potential. And so in the next web lecture, we're going to take a closer look at what happens once this threshold is reached and what causes this extreme change in the polarization of the membrane that we call an action potential, and also what happens at the end of the axon once that action potential reaches the end, how it communicates that message to the next neuron.